Researchers at the Diabetes Research Institute urged Biorep to come up with a better way of doing cell secretion analysis. Since then, we have been determined to deliver the best tool on the market. Now in its fourth version, our PeriFusion system has evolved into a high-throughput, automated robot capable of processing multi-plate protocols without supervision. Sources are automatically selected and distributed on the fly by an automated manifold and delivered by a precision multi-head peristaltic pump to up to 12 sample chambers in parallel. All reactions take place in a temperature-controlled incubator while cell secretions are automatically collected on refrigerated well plates. The system is controlled through an intuitive touchscreen interface and, with the optional orbiter, it can handle extended unattended operation. Accurate, repeatable, and fast, the Biorep PeriFusion system sets a new standard in cell secretion analysis. Following the arrival of your new PeriFusion system, please ensure you have received all of the equipment and accessories. PeriFusion chambers, replacement O-rings, filters, polyacrylamide bead suspension, dispensing nozzles, pump cassettes, PeriFusion tubing set, source tubing spool, cooling tubing spool, cell loading rack, tubing clips, standard and deep 96 well plates. Connect and power up the machine. Although the machine has a touchscreen, you can connect a USB mouse if needed. Wait for the machine's screen to boot up and press anywhere to begin. To use the PeriFusion system, you must have a user account. All settings and protocols will be saved on your account for future use. Click Add User. Type a unique username. Choose a password and click Save. You can now select your account on the drop-down menu. Type in your password and press Login to continue. On the main menu page, you can choose to start a protocol, configure the machine settings, or test subsystem functionality. You can also home or park the motion system, or you can finish a session by logging out or shutting down. Let's start by testing the subsystems. On this screen, you will have the ability to manipulate and test each automated component independently. Test the motors and limit sensors of both axes of the motion system. Check the tray orientation sensor is functioning properly. Now test the main pump. On. Wait. Off. The tray pump. On. Wait. Off. And actuate the valves, 3 or so consecutive on-off, of the fluid handling system to check they are working fine. Perform a quick test of the heater and fans of the temperature control system. On. Wait. Off. And finally, test the alarm buzzer. All systems check. Enter the Configure Settings page to specify the well plate type and handling method if equipped with the optional orbiter robot. You can specify which temperature alarms you want to be active during your experiments. You can also activate advanced options such as the Action Required Alarm or Skip First Row. You are now ready to start an experiment. To perform an experiment, click on the Protocol wizard. The first step is to select the number of chambers to be used in the experiment. We will use 4 chambers in our example. Note that the position of the chambers on the screen should match the final position of the chambers on the machine. Press the right arrow to continue to the next step. In the protocol page, you can easily create a new protocol or select a previously saved one from the drop-down. For our demonstration, let's create a new protocol. Click on the New button. Click on the protocol name field and give it a unique name. Add step number 1 by clicking on Add Step. Now replace the X placeholders with the step parameters: 10 reps, 10 seconds for each rep, at a 200 microliter flow rate. Now let's assign solution number 1 to channel A, solution 2 for channel B, and 0 for C and D, since they will not be used. The last column is an advanced option that will add a pause before the step begins. We will not pause in our experiment. Click Add Step to add the next step. Fill in the parameters as before. Continue to fill in the steps until your protocol is complete. Click Save. You can see the new protocol is now available on the list. The machine automatically calculates the number of 96 well plates needed to complete the protocol. If the tray is switched from portrait to landscape, the machine will immediately update the number of well plates required. Move to the next step when ready. Follow the on-screen instructions to set up the chamber. You may be able to skip some of these steps as we continuously make efforts to simplify the process. Place a circular fiberglass filter inside the bottom cap bore. Place an O-ring on top of the filter, making sure not to wrinkle the filter. Tighten the bottom cap on the chamber body. Place an O-ring inside the chamber body bore. Connect the short segment of the perifusion tubing set to the outlet tube of the bottom cap all the way up to the plastic. Repeat the process for all the chambers involved and set them aside on the auxiliary rack. Click Next to continue with the tubing setup. Follow the on-screen instructions to set up the tubing. We are constantly striving to simplify setup steps, so you might be able to skip some of these steps. Begin by setting up the source solutions. You can use up to 8 different sources on the removable tray. We will be using 3 in our example. Connect each source to the corresponding input port on the valve manifold with a section of the source tubing spool. Insert the 3-stop segment in the pump as follows. Slide the tubing through the cassette groove. The orange-colored detent on the manifold side should mate with the notch in the back of the cassette. Stretch the tubing enough to lock the green detent with the notch on the lever side of the cassette. When the tubing is secure, insert the cassette and push until the lever locks in place. Repeat the process for all remaining sets. Manifold output. Connect the free end of a pre-assembled perifusion tubing set to each output of the manifold being used. The other end should have a chamber top in place. Repeat the process for all remaining sets. Click Next to continue to the next step. The system will automatically create a priming sequence based on the selected protocol. Choose a prime flow rate and step time. Place a paper towel on the tray and press play to start the sequence. The manifold will cycle through all the solutions, removing all the air from the system. Priming will stop automatically when complete. Press Next to continue. With the system primed, the cells can now be loaded into the chambers by following the on-screen instructions. The bottom halves of the pre-assembled perifusion chambers should be on the cell loading rack. The chamber top should be hanging from the tubing set above the waste bin. Using a pipette, add 100 microliters of the bead and buffer suspension to each chamber. When fluid is observed at the output tubing, clamp the tubing to retain the solution. Add the cell sample. Add another layer of the bead suspension, making sure to top off the chamber. Start the pump slowly at approximately 100 microliters per minute until solution starts dripping from the top caps. Screw each chamber to its corresponding cap by carefully turning the chamber body and quickly removing the clamp from the bottom outlet. Stop the pump. Place each chamber in its designated location in the machine rack and the stainless steel nozzle in its corresponding well. Click Next to continue. Usually, cell secretions collected on the well plate must be cooled for preservation. Connect the sample tray to a coolant reservoir as shown. Both the cells and the stimulant solutions must be at the specified temperature. Close the machine cover and enable the heater. This will bring the enclosed environment to the set point within a few minutes. Prime the pump with coolant and turn the cooling pump on. When the present value of the incubator reaches the set point and the tray has cooled down, you are ready to perform a perifusion experiment. Click Next to continue. As its name suggests, this page is used to monitor the machine parameters during a perifusion experiment. To begin, press play once. The machine will home and the protocol parameters will be filled in. If everything looks okay, load a 96 well plate and press play again to begin. You will see the protocol step information to the left, the valve activity map in the center, and the temperature readings on the right of the screen. Other information is available, such as elapsed time, alarm status, well plate type, and special conditions, such as skipping the first row. You can sit back and relax. The perifusion system will do all the heavy lifting, dispensing, switching, and moving according to the protocol. The machine will alert you when it needs your attention, such as changing a full well plate. When equipped with the optional orbiter robot, the perifusion becomes a high-throughput machine, capable of automatically handling extended protocols spanning up to 20 standard well plates. The orbiter will switch well plates in less than 10 seconds and store the used one in a refrigerated rack for preservation of the analytes. An orbiter-equipped machine will have the following extra features: a green Enable button in the front panel and an emergency stop for safety. To use the orbiter robot, press the green Enable button and switch the orbiter power toggle in the Settings page. Before using the orbiter, remember to select the type of well plate being used between Deep and Standard on the Settings page. If your experiment uses a standard well plate, remember to insert the adapter. Now select the tray orientation to be used, landscape or portrait. According to these choices, you must load the well plate rack carefully, making sure that the A1 well is positioned properly for all the trays in the stack. If the experiment uses a portrait configuration, the well plates must be loaded as shown. If a landscape orientation is used, the well plates must be positioned as shown. The orbiter is the ultimate perifusion add-on for extended unsupervised operation, allowing you to use time more wisely. After the protocol is complete, you will be able to generate an automatic report in the following screen. Give the experiment a unique name. Fill in the number of chambers assigned to each output channel. In our case, there were 2 chambers on the A channel and 2 chambers on the B channel. Assign a name to each of the solutions used in the experiment and identify it with a specific color in the key. For example, we'll name the first solution S1 and give it a green color, the second solution S2 and choose a blue color, and so on. When ready, press Generate Report. You will see the report being generated on the screen. The report will contain all the information relevant to the experiment, a temperature graph of the incubator, and a well plate map showing the stimulant solution used for each well. When done, just close Excel, maximize the perifusion, and continue to the cleanup page. After every experiment, the valve manifold must be cleaned thoroughly to maintain optimal performance. Impurities left in the manifold can damage valve parts over time, impairing proper protocol execution. The last page of the Perifusion Wizard will guide you through the cleaning process. Change all the sources for deionized water. Place a paper towel on the tray and press play to start. The machine will automatically cycle through the used valves to flush out any residue. The machine will prompt you to change the source for a 1:9 bleach solution for sanitation and again with deionized water. Finally, the machine will prompt you to remove all sources. This will draw air into the system, removing the water in the manifold. When the cleanup process is complete, you can press the Biorep logo, which will bring you back to the main menu. Before shutting down the machine, press Park so that the tray is stowed away, preventing accidental damage to the motion system. To shut down, press the red power button and confirm. You can switch the main power when prompted. the Biorep perifusion system, the standard in cell secretion analysis.